In the session, we're going to be looking at infection and response. We have got content here that's covered in all specifications. If your exam is highlighted in green, it means that you could be assessed on this in your exam. While if it's in grey, this is content not on your specification. So I'd suggest find the key for your exam. So Combined Science Foundation is here, Combined Science Higher, Separate Science Foundation and Separate Science Higher, and track these through as we go. I'll flag it up during the video as well, but if these are ever in grey, it means you will not be assessed on it. But if it is in green, it could come up in your exam. We'll start with looking at communicable diseases, and this is a really common definition that you might get asked within your exam paper. And communicable diseases are ones that are caused by pathogens, and pathogens is a microbe that can cause infection in an organism. So communicable diseases are caused by pathogens and they're contagious. They can be passed from person to person. And examples of pathogens can include viruses, bacteria, protists, and fungi also. In different ways, these can be passed on. And as we go through the content for today, we'll look at some different diseases, what causes them, how they spread, and how we could maybe prevent their spread also. But the idea about communicable diseases are these infections that you can catch and can pass from person to person. But again, as that definition, if you're ever asked what a pathogen is, this is a microorganism that can cause infectious disease. There are different ways that disease can spread, and a few kind of visual hints as we go through these, but we'll take each in turn. The first is direct contact. So this is example of touching someone's skin, kissing, sexual contact, but also leaves of plants touching a diseased leaf of another plant. So they're all having to come into direct contact to spread that infection. We also have infections that can be spread by air and by droplets. Now droplets tend to be dispersed in the air, but they are slightly different. So with droplet transmission, this is when someone coughs or sneezes, and what they'll do is disperse droplets of water within the air. But within those droplets of water, we will have the microbe within. And it means that if we inhale this, we're then at risk of contracting that illness as well. So droplet is when we have someone coughing and sneezing, dispersing tiny bits of water into the air, and that could be inhaled by someone else, which then will spread that infection along. With air, this is when pathogens, instead of being carried by water, are more carried by dust. But within the air, we can get droplets suspended in too that can cause infection. Plants, for example, their infections can often be blown from the soil by the wind and used to spread from one plant to the other. We also have infections that can be spread by water. And typically what we'll find is that microbes in dirty water, so sewage or maybe water that's been contaminated with sewage, can then cause disease in plants and in animals. We also have microbes present in our food that have the potential to cause infection as well. So what we typically find is that food's not been stored correctly, it's not been cooked correctly, or just not kept in the most hygienic conditions. We can have bacteria at a high enough level on our food that can cause infection. And we'll kind of touch on this when we talk about salmonella a little bit later through. We also have transmission by vectors, which is a bit of a strange term, more unfamiliar to students, but it does come up on both paper 1 and paper 2 when you look at genetic engineering. But for now, for your paper 1 content, a vector, in this case, is an organism that will transmit a disease to another organism. So it's one organism that will spread a disease to something else. So for example, mosquitoes can spread malaria. They don't suffer from malaria themselves, but they can spread it from human to human. And rats were also responsible for plague, but again, they weren't suffering from it. So these vectors are organisms that can transmit a disease to another organism. But mosquitoes and your rats are a really good example of this. Now, another common question that comes up, and it's often a 2-marker, is you're asked why pathogens make us feel ill. And what we typically see is the first mark's answered pretty well, the second mark can get a bit weak. So with pathogens, what they're going to do when they enter our body is they're going to reproduce incredibly rapidly. And as they do so, they're going to produce toxins, so poisons, that damage our cells and are going to make us feel ill. So if we're thinking about the breakdown of the 2 marks, you want to say that they're going to reproduce quickly and that they're going to produce toxins that will damage us or damage our cells or make us feel unwell. But it's about the production of those toxins that's going to make us ill. But there's a lot of the toxin because the microbes reproduced really quickly. Now, viruses have got another hit, and they make us feel particularly ill because what they actually do is they invade our cells and damage our cells on top. So it does mean that viruses, you've got an extra point. So we can talk about the fact that they'll enter our cells, reproduce rapidly, and produce toxins. But in doing so, they are going to be damaging our cells because they invade cells as part of their life process. Now, for your specification, you need to be prepared to discuss some different infections, what causes them, and how they spread. So our first one is a viral infection known as measles. And this is typically seen as a fever and a red skin rash. Now, there are fatalities with this, and it can lead to complications that can cause death. This is spread by inhalation of droplets from sneezes and coughs. In terms of the prevention, if someone is suffering from measles, the best thing to do is isolate them from anyone who might be at risk of catching that infection from them. So at that point, isolation. But in terms of controlling the spread, our big measure and prevention of this is vaccination programs. So measles is in the mumps, measles and rubella vaccination, so the MMR. Because viral diseases are particularly hard to treat, and so the only best thing we can really do is control their spread in the first place. And that's why vaccination is so important. But if you have someone who's suffering from measles, then sensible ways of preventing their spread would be, right, you'd isolate them until they're feeling better. You'd make sure that people come into contact with them was wearing a mask and washing surfaces to prevent them contracting this through the inhalation of the droplets. Another viral infection is HIV, and this initially causes flu-like symptoms and is spread through the exchange of bodily fluids. So it means it can be sexually transmitted or contamination from needles. The controlling spread here is through, not sharing needles, and we've got barrier methods of contraception also. There are some additional points that fit into the specification that are worth being aware of. So later stage HIV is known as AIDS, and this is when the body's immune system is damaged. And it means that that immune system is no longer able to effectively deal with minor infections like colds or actually picking up cancer cells that might be in the blood. But we can use antiviral drugs that can slow the development of HIV into AIDS, and so almost delay that onset of the actual issue that will come about here with the issues with the immune system. The final virus is a plant virus known as tobacco mosaic virus, or shortened to TMV. And this typically causes a discoloration of the leaves of plants. Now, this is spread through direct contact, so that might mean leaves touching each other, but it also might mean like a gardener using like secateurs on one and then accidentally touching another and passing it on that way. The only way really to control the spread of viral infections in plants is dig up and destroy the infected plant, because all of those leaves are going to come into contact with possibly other plants. That's the only way of really controlling the spread there. But also any equipment that's kind of been used around those plants and the hands both need to be washed because of the fact this is spread by direct contact. Now, the most likely way that you might get an extended question within this is actually being asked how this virus might affect the growth of plants. And it does kind of overlap with photosynthesis a bit later on in your specification, but it is worth being prepared for it within your exams. So with tobacco mosaic virus, you're going to find a discoloration of the leaf. That will cause a reduced rate of photosynthesis. As a consequence, that will mean that we've got less glucose produced by that plant, which means we have less glucose for respiration, so we're going to have less energy. And that might pick you up 3 marks within an extended exam question. The bit to be aware of is that also glucose is not just essential for respiration and energy release in a plant, but glucose is also really vital in making amino acids. So the second issue that you need to discuss with this is that if we've got less glucose, we're going to make less amino acids, and that will mean we make less proteins. So it's almost like a chain of events to learn here. The consequence of that is we'll get less growth. So the plant has got the discoloration of the leaf, reducing photosynthesis and reducing the amount of glucose made. But then there's 2 issues with that reduced glucose. We've got less glucose for respiration, so less energy, but also we've got less glucose to make amino acids, to make proteins as well. Both of those, less energy and less protein, is going to result in less growth. So typically this plant will be stunted. It's not going to grow as big as otherwise it should. On to bacterial infections. Our first is Salmonella, and there are quite a wide variety of questions that have been assessed around this in the past, and it's normally about controlling spread. Now the symptoms of Salmonella are fever, abdominal cramps, vomiting, diarrhea, and this is all down to the toxins that have been produced by the bacterium that have infected the body. One thing I've noticed in the exam questions is they might, for example, give a symptom and ask you to give a couple more. When in doubt, fever in animals is normally a quite good one to give. This is spread through contaminated and undercooked food. And we do find this bacterium present, for example, in particular on meat. One way that we can do prevention here is that poultry can be vaccinated against Salmonella. What we can also do is regularly test the poultry, and if we find that there is a Salmonella outbreak within the poultry themselves, culling them. So actually destroying those chickens and not taking them forward to be consumed would be a way of controlling the spread. However, in the home, it comes down to importance of cooking food properly, making sure that surfaces have been cleaned, hands have been washed, and that meat has been stored correctly. All of those things are going to control that spread. So that's our Salmonella. Gonorrhea is another bacterial infection. Now with this one, it causes a thick yellowy greeny discharge from either the vagina or penis, and also you're going to have pain when urinating. And this one's spread through sexually transmitted. In terms of controlling spread, this is down to the fact that using barrier methods of contraception to actually prevent the spread from one person to another, but also we do have antibiotics that can treat this infection, and so also if someone is suffering from the infection, is to have an antibiotic course of medication to clear the infection up. One possible link that I've noticed with AQA is that they have started to make links to antibiotic resistance within gonorrhea, and starting to link in, well, how has this occurred? Or why are there concerns around this? So with antibiotic resistant strains, these are now gonorrhea infections that we do not currently have antibiotics that are effective against it. They have become resistant and are no longer destroyed by penicillin, the drug that typically in the past would treat this. On to the fungal diseases, we've got rose black spot, and this is where we get, again, discoloration of the leaf, but we get these like mottled dark spots that appear over the leaf itself. Now, this is now going to be spread by both water and wind, and the only real way to treat this is to remove and destroy the affected leaves, and that means even those that have fallen off the plant already, you don't want those going into the soil, and you can use fungicides to treat the fungal infection. Again, you might be asked how rose black spot affects growth, and you'll notice that this is a very similar answer to tobacco mosaic virus. We have got a discoloration and loss of leaves causing reduced photosynthesis. This means less glucose is made, therefore less respiration, less energy. But we've also got less glucose to make amino acids, so we'll make less proteins. Both of these things are going to cause, as a consequence, less growth. Our process disease is malaria, and this is one that is caused by a eukaryotic microorganism. So these have a nucleus, and actually they are a group of pathogens that do have a nucleus, and these are protists. Malaria is the one example in your specification of an infection that's caused by a protist, but the mosquito acts as a vector and spreads this from one infected person to someone else. So what happens here is the mosquito will feed on the blood from an infected person, and when they then go to feed on the blood of another person, can inject that vector into the bloodstream of that next person. The mosquito is not made sick with this at all, but it is acting as a vector and spreading the disease. So this malaria then is caused by a protist, and we find the infection spread where the insects live, so where we're going to find the mosquitoes. So it's in the southern hemisphere of the world. One common question is about how we can prevent its spread, and there are two aspects to this. One, how can we stop ourselves being bitten by a mosquito? And two, how can we actually deal with the mosquitoes? So in terms of being prevented from being bitten, we can use, they've used in the past mosquito nets when people are sleeping. It's about wearing protective clothing. You might avoid going out at times when mosquitoes are most active. One thing to be really careful about is how you refer to insect repellent. It's come up in the past within exam questions, and just be aware you need to use the word insect or mosquito repellent, not mosquito spray. That would not be accepted within the exam because it could be inferred from your description, if you said mosquito spray, that you'd be spraying mosquitoes out of a can. That would not stop malaria. So you have to be really careful about how you phrase that one. So mosquito repellent is fine, but mosquito spray is not. Be really accurate with how you're going to describe that. What we can also do is prevent the mosquitoes themselves from breeding, and they breed in shallow waters, so still standing water. If we drain those and get rid of the areas in which they breed, that can reduce the number of mosquitoes in the general area. The other thing that we can do, again, in areas where we know they're going to breed or be found, is we can actually spray insecticides. Malaria then causes fever, sweats, chills, headaches, vomiting and diarrhea, and can cause death. It is spread by mosquitoes that are acting as a vector, and we can control their spread by either preventing people from being bitten, so that might be using mosquito nets, mosquito repellents, protective clothing, avoid being out when we know they're most active, but also we can prevent the mosquitoes from breeding. So we can remove and drain any stagnant, still water, and we can spray insecticides on those areas too. There are antimalarial drugs that can be taken, and they can treat the symptoms, and they can also prevent infection. What we do find is that malaria is found in areas of the world with high temperatures, and it is responsible for killing a huge amount of people per year. Moving on then, we're going to have a look at what our defences are against these infections. And we've got a series of different defences within the human body. We have physical defences, so these are barriers that prevent microbes from entering, and we also have chemical defences. So some common ones to be aware of is our skin, first of all. We have got a dead layer of skin that's really tough to kind of penetrate, acting as a barrier. And we also find it secretes these antimicrobial chemicals, which is able to kill some microbes on our skin. If we get cuts, we're going to see that we can form scabs that can stop further microbes from entering. We've got hairs within our airways, And those, for example, on our nose can be used to keep dust and some microbes out, but our mucus and cilia in our trachea and bronchi are present also. So the mucus's job is to trap the microbes, while the cilia, these fine little hairs on the cells, their job is to move the mucus along and clear it out of the airways. Our chemical defense is our stomach acid. So if we do swallow any food that's got microbes present, we can find that the low pH of our stomach can kill off some, but not all microbes. Once an infection enters our body, we have a second line of defense, which is going to be our immune system. Now, when anything infects our body, any pathogen has this little protein known as an antigen that covers its surface. And this is the marker for our body of this should not be in our body. And it triggers an immune response. And the antibodies can attach onto these. So this will kind of trigger and alert the body to something's not quite right and needs to be addressed within there. So these are foreign proteins that will cause an immune response. The immune response has got three actions. We have got phagocytosis, produce antibodies, and produce antitoxins. So three things will start to happen at the point which our immune system acts. Phagocytosis is when the white blood cells engulf and ingest a pathogen, causing it to be destroyed. Producing antibodies is a way of actually supporting phagocytosis in a way. What we'll have is antibodies are produced and they will bind onto the antigen, causing the microbe to be demobilized, so it will stop moving and it will clump together. And then hopefully a white blood cell will come around and carry out phagocytosis, engulfing those demobilized microbes up. Now, antibodies will only work against specific antigens. So it does mean that one antibody will only work for particular diseases. The final action is they'll produce antitoxins, and these are chemicals in the body that are designed to counteract the poisons, the toxins that the pathogen has made. Vaccinations are a way of stimulating an immune response to actually provide protection for us later on in life. With a vaccination, what we typically do is we inject something such as a weakened or inactivated or even part of the pathogen into a person. But it's not anything that's going to cause disease in that person. So there's no issue of getting the illness, but what it will do is cause an immune response. Vaccines work then by they contain an antigen for the pathogen. We inject this into the body, and when we get reinfected, the body can very quickly make antibodies effective against that invading pathogen. So upon reinfection, we make a larger quantity of antibodies and we make it much more rapidly than we would on the first infection. What this means is that the infection or the response to the infection is so quick and so rapid that we don't actually suffer from the infection. And as a result, you're not going to feel ill. Now, the pros of this is we're going to prevent people becoming ill. Our cases of measles will reduce if we've got a higher number of people vaccinated. We get this idea of herd immunity. So that idea that maybe there are people in the population who can't be vaccinated, they'll be protected by the people around them who are vaccinated, unable to transmit the disease. It's also cheaper to vaccinate against the disease in some cases than it is to actually treat it. However, the problem is that there are known side effects sometimes to the vaccine, and so they do come with side effects attached. There has been some controversy over the MMR after there were false claims made that it caused autism. And what it did mean is the rates of vaccination for the MMR decreased. And remember, MMR stands for mumps, measles, and rubella. So what we find is that when we have our first vaccination, we don't have any of the antibody or a very small amount of it in our blood. But after our initial exposure of that antibody, we will gradually produce the antibody, which will then kind of decrease. But upon re-exposure, so let's say this person has now come into contact with the actual pathogen, what we'll find is that the response to this is faster. So the delay in actually starting to make that antibody is less, and they'll make a much higher quantity of that antibody. As a result, that person is way less likely to become ill. And it's all down to the fact they're responding quicker and to a higher degree with that antibody production. We do have a range of drugs that come up within specification. First of all, antibiotics are medicines that can cure bacterial diseases. Penicillin is one example of this, and they damage the bacterial cells but do not damage our body cells. They cannot, however, kill viral cells because viruses are inside of cells. So antibiotics can't damage our cells, which means they definitely can't get to anything inside them. So if viruses are infecting us, antibiotics are not going to work. What we do find is that certain antibiotics only treat certain bacterial infections. So there isn't just one antibiotic, there's a huge range of them. But when doctors are giving these out, they need to consider the side effects, how effective that drug is, the cost to the patient and to others, and the benefit. So it does mean for mild infections, you might not be given an antibiotic because your body can get better on its own. The other concern is antibiotic resistance. What we find is this idea that there's a kind of variation within any population, and that includes bacteria. Some will be slightly more resistant to the drug than others. So when someone takes the antibiotic, what can happen is that we've got loads of bacteria, maybe some are a little bit more resistant. The antibiotic is taken, and the ones that are less resistant die first. If that person then stops taking the antibiotic, the resistant one has survived, and it's now in an environment with no competition. That bacterium will now reproduce, and it will actually produce a load of bacterium that are also antibiotic resistant. And this can carry on again. So we find this variation within the population, and some are just more likely to survive than others. What this does mean is the effectiveness of our antibiotics is starting to decrease. We now have certain bacterium that are resistant to the antibiotics that once worked against them. The concern around this is that this tends to be the mutations are always happening faster than we can produce new drugs to treat them. And so one thing that can often be asked in the exam is about how we can prevent this. So on the doctor's level, it's about making sure that they're not prescribing for non-serious infections, and that they are definitely prescribing for bacterial infections and definitely not virus. On the patient side of this, it's about the fact that they need to continue to take those antibiotics as they've been instructed for their entire course. So the doctor's role is to make sure that they will not prescribe these unless it is a serious infection. So infections where you can naturally just get better on your own, the idea is you would not be prescribed antibiotics, and it wouldn't be given for a viral infection at all. So that's the role of the doctor. While for the patient, it's about the case of you finish your course of antibiotics, even if you're starting to feel better. Even when you are starting to feel better, there will still be some bacterium in your system, and you want to make sure they're completely gone. The other round of drugs is painkillers, and these do not treat infections but can reduce the symptoms and make people feel more comfortable. Now, there are some drugs in the specification that have been around for quite a while, and they're often one-mark factory recalls. So Foxglove contains a drug known as digitalis, and has been known to strengthen heart contractions. Willow contains a drug known as aspirin, which has been used as a painkiller for a long time. And we also have the fungi known as penicillium that produces the drug known as penicillin, and we use this as an antibiotic, and this was discovered by Alexander Fleming. Most new drugs though nowadays are made in the pharmaceutical industry, and we go through drug trials. Now this tends to be, or could be, some extended questions. With this, be aware of the different stages and what's involved. So we've got preclinical. This is where we test animals or cells and the purpose of this is to test for toxicity. We then go into phase 1 clinical trials. Here we test small numbers of healthy individuals and we test for toxicity as well as side effects. So we're looking for the toxicity or side effects of this. Now we use low doses here for safety. We use healthy individuals first of all. We're not looking to see if the drug works, we're just looking to see about the toxic side effects. Now we use healthy people also because they're less likely to be taking drugs that might interact with the drug they're being given, which might make it harder to see the side effects. Once we're satisfied this is safe, we then move on to our phase 2 clinical trials. This is where we test on a small number of patients, and the purpose of this is to find the right dosage, to see if the drug works, otherwise known as efficacy, and to test for toxicity. A few extra details around this. We normally do this as a double-blind trial, so neither the patient or the doctor knows who's got what, and that's to avoid bias. We also tend to use a placebo that's a dummy or fake drug that does not contain the drug at all. And the idea here is if we see if actually the drug is making an effect over doing nothing at all. Whenever we do these trials though, it is important to make sure that we try and control everything else as much as we can. So the age, the gender, for example, of those people. We then move on to phase 3, and the only difference here is it's now in a larger number of patients, but we're still looking for dosage, efficacy, and side effects. Once that's done, we then move on to peer review, and this is where the trial itself is tested. Now, it's a really common 2-mark question to ask why we do this. 2 key phrases to use is to avoid bias and to make sure the results are valid. So avoiding these false claims that could be made. When we're using these drug trials and using placebos, there is an ethical implication of that because these are dummy drugs. They do not normally contain active ingredients. What that does mean is that if we were maybe testing a drug on a deadly disease, so like cancers, for example, you wouldn't want to be in a position when you're in a placebo group not receiving any treatment at all. And so often what we do for this is we use a drug anyway that's already currently on the market. So sometimes when we're looking at diseases that could actually kill if someone's not been given treatment, we might choose instead to be like, right, we'll give the new drug we're testing and we're going to test it with another group of people taking a medicine that already exists to treat this disease. We're going to now move on to the separate science only content, and there's a couple of things we're going to look at: monoclonal antibodies and plant disease. With this, monoclonal antibodies are identical copies of one type of antibody. They are man-made proteins, and you need to be prepared to talk about how they are made. And they are made from a single clone of cells. They are antibodies, so it means they are specific to one kind of antigen. In terms of their production, there's some really important key phrases to use. First of all, what we'll tend to do is isolate the antigen that we want to make an antibody for. Once we have that antigen, we'll inject it into a mouse, and the mouse's lymphocytes are stimulated to produce antibodies. We then harvest those lymphocytes and we combine them with tumour cells, and this makes what we call a hybridoma. So the lymphocyte is what's producing the antibodies. The tumour cell will divide very rapidly, which is an ideal trait that we want in this future cell. So we've got these hybridomas produced. The next piece, and we need to be very careful in how we phrase this, is we are going to isolate the hybridoma that's making the monoclonal antibody that is specific to the antigen we want to target. So that's the one that I kind of stress, be really careful and particular about how you word that in your exam. What we'll then need to do is clone the isolated hybridoma, which will happen really rapidly, and they will make antibodies. And those are the key phrases or stages involved. At that point, we'll then harvest those antibodies and we'll purify them. So monoclonal antibodies are man-made and they're specific to one binding site. A hybridoma cell is a lymphocyte and tumour cell combined. Our uses for this include lab tests for pathogens. It includes lab tests for testing chemicals in blood. It also means we can locate certain molecules. We can use it in pregnancy tests. We can use it to measure levels of hormones, and we can use it to treat cancer. Monoclonal antibodies have been used to treat this with cancer in particular, we bind on a toxic substance to a monoclonal antibody. That will then specifically bind to the antigen, and at that point, when that binds onto the cell, it can deliver the drug and either kill the cell or stop that cell dividing, either of which it would in theory treat that cancer. We can also use it in diagnostic tests. Now in this, we are now attaching a fluorescent dye to the antibody, and with this, it will then make this visible, and we can look at that under a microscope or on like a strip test also. So when we're looking at monoclonal antibodies in diagnostics, we'll attach on a fluorescent dye, and it's then actually talking about that when this then binds onto the antigen, it will make that substance that had the antigen become visible. Now the pros of this is there's loads of different uses that we could in theory use this for, and we could design these antibodies to bind on to pretty much anything that's got an antigen. It could be used to treat cancer, and we also get very fast sensitive tests. So for example, they're used in pregnancy tests. However, the problem is that we're using animals to produce this, and there's ethical concerns around that. They're also pretty expensive to make, and also there's more side effects than expected. And be prepared, you might maybe be given data to evaluate a different procedure using these monoclonal antibodies. The last section of this is looking at plant diseases. Now, these can be detected, and that's the idea of how looking at a plant you realise that it's ill. With this, you might see spots on leaves, you might see areas of decay, you might have these weird growths forming on it, the stem or leaf just looks a bit of a funny shape, even pests are present, and we could see discoloration of the leaf as well as stunted growth. So those are things we might see on a plant indicate it might be ill. The identification is how we then actually narrow down what it's sick with. So here we can use referencing, using guiding manuals or websites. There's even apps now that do this. We could take it to a lab to identify the pathogen, or we could use monoclonal antibody testing kits. So be prepared, there's an application there they could pull across. Now we've talked about TMV and black rose spot earlier on. We also have aphid infections too. Now with our aphid infections, these aphids have a mouthpiece that's able to pierce into the phloem of the plant, and they'll feed off the sugar within. Now with less glucose, it means that we're going to have less respiration and less energy, and we'll also be able to make less amino acids, therefore can't make as much protein. We will see stunted growth of that plant. There are a couple of deficiency diseases to be aware of too. So nitrate deficiencies stunt growth because nitrates are needed to make proteins. If we've got less nitrates, we can't make amino acids, we can't make proteins, there will be stunted growth. Magnesium is needed to make chlorophyll, and if we get a deficiency, it's known as chlorosis. Now the plant will have less chlorophyll for photosynthesis, so therefore we'll have less glucose, and it's back to that writing pattern of less glucose, we're going to have less respiration, less energy. We'll also have less amino acids made, so less proteins made, so less growth there too. So magnesium deficiency in particular has got a bit more that you can kind of discuss around this. Plants do have some defences, and it's not just humans that have them. So plants are able to defend themselves against infection, but also been eaten too, or trampled inadvertently. So plants can have physical defences. This includes their cell walls that are incredibly strong and tough. They also have these tough waxy cuticles on the outside of their leaves. And actually on their stem, their bark is dead. It can fall off and actually discard the pathogen that maybe has infected it. There are also chemical defences. So some plants can have poisons, and that's mainly due to herbivores. It won't prevent from infection. But some plants, like peppermint as well, sage, they can have antimicrobial chemicals that prevent bacterial or microbial growth. So these kill bacteria that might maybe get through those physical defences from earlier. There's also mechanical defences where we've got thorns that can deter animals. We also have sensitive plants, so their leaves can droop or curl when touched, and mimicry. The purpose of this is normally to look unappealing so that something's not going to want to eat it.